Watch this physical action. A thumb rhythmically striking a glass pane, refreshing a feed for the 50th time in an hour. Observe the user's face. There is no smile, there is no laughter, and no visible sign of satisfaction. We are looking at a state of extremely high motor engagement that yields exactly zero hedonic payoff. Popular culture frames this behavior as a lapse in willpower or a psychological weakness, but the clinical data tells a different story. This is a measurable structural failure in the brain's biology. We are watching an organism's ancient survival architecture being hijacked, forced to endlessly execute a sequence of actions that no longer provide a reward. To understand exactly what is happening here, we have to audit the hardware. The core thesis is this: the neural network responsible for desiring a stimulus has been surgically severed from the network responsible for actually enjoying it. The site of this disconnection is a highly specific sequence of structures known as the Mesocortico-limbic projection pathway. Tracking this glitch requires us to strip away subjective psychological narratives. We need to examine the raw mathematical and molecular evidence. This map shows the Mesocortico-limbic circuit. It originates deep in the midbrain, inside a structure called the ventral tegmental area. The ventral tegmental area acts as the brain's motivational engine. It synthesizes dopamine to generate a state known in neuroscience as incentive salience, which translates directly to the sensation of wanting. In this system, dopamine operates as an allocator. It tags neutral environmental cues, like the ping of a notification or a red app badge, and turns them into motivational magnets that compel your attention. Here is the baseline state. This meter displays the steady tonic firing rate of dopaminergic neurons, ticking at 5 Hz. This sets your baseline arousal. But the exact millisecond the brain detects a predictive digital cue, that pulse violently spikes to over 50 Hz. This is where we must correct a widespread misconception. That dopamine spike does not encode pleasure. It exclusively encodes the pursuit of utility. The wanting machinery is massive, robust, and functioning exactly as designed to drive relentless pursuit. To find the actual sensation of pleasure, we have to look away from this sprawling dopamine network and drill down into microscopic subregions of the nucleus accumbens. These are the hedonic hotspots. They are the structures responsible for hedonic impact, or liking. This extreme macroscopic zoom reveals those regions, highlighted here in surgical blue. Unlike the dopamine network, pleasure here is mediated by entirely different neurotransmitters, primarily mu-opioids and endocannabinoids. Notice the anatomical fragility. These hotspots are incredibly restricted in size, barely 1 cubic millimeter in volume. True pleasure relies entirely on these tiny, fragile microzones. Because of their structural limits, the biological hardware is completely incapable of sustaining the high-frequency demands of modern digital environments. Under continuous bombardment from digital stimuli, watch how these localized receptor clusters dim and shrink. The fragile opioid hotspots rapidly undergo desensitization and tolerance. But the massive dopaminergic system reacts in the exact opposite way. The wanting network thickens and sensitizes. It becomes hyper-reactive to cues, even as the capacity for pleasure drops out from underneath it. The academic literature is definitive on this decoupling. The brain's wanting and liking systems are produced by separable and independent biological mechanisms. Through the process of sensitization, repeated digital triggers cause the mesocortical limbic circuitry to become aggressively overreactive to reward-related cues. We do not have to guess that this is happening. Using advanced clinical imaging like PET scans, we can physically measure this structural shift in human patients. The quantitative data is stark. Chronic digital overstimulation causes a 20% downregulation of D2 dopamine receptors in the striatum. This precise level of receptor reduction mirrors what is observed in patients with severe substance abuse disorders. The decoupling is complete. The brain is now structurally starved for a baseline level of satisfaction it can no longer reach, forcing the user into a state of continuous, joyless pursuit. To understand why the brain keeps pursuing a reward it no longer enjoys, we have to look at the computational engine driving the dopamine network. Reward prediction error. This oscilloscope visualizes the temporal difference equation. Dopamine neurons calculate the discrepancy between a predicted value and the actual reward. Under normal conditions, when a reward is fully predicted, the error hits 0. Notice how the signal flatlines. The brain habituates, and dopamine shuts off. Digital interfaces are engineered to bypass this habituation. By utilizing variable, probabilistic reward schedules, they ensure that the predicted value never perfectly matches the outcome. This exploits a specific biological asymmetry. Because dopamine neurons idle at a low firing rate, negative prediction errors are trapped by a hard biological floor of 0 Hz, but positive errors can spike upwards, unbounded. By delivering random validations, the interface mathematically forces the average prediction signal to drift upward. It locks the mesolimbic system into a perpetual state of positive prediction error, trapping the user in an endless loop of anticipatory craving. The brain cannot sustain this state of perpetual dopaminergic overheating. To protect itself, it initiates a drastic homeostatic defense. It recruits the lateral habenula, a deep brain structure that acts as a powerful emergency brake on the dopamine system. The lateral habenula floods the surrounding neurocircuitry with an endogenous opioid called dynorphin. This chemical plunge induces a state of persistent dysphoria and anxiety. At this stage, the individual's primary behavioral driver has fully shifted. They are no longer operating on positive reinforcement to seek pleasure. They are operating on negative reinforcement, compulsively engaging with the screen merely to escape chemical misery. When attempting to solve this, the most common internet protocol is a dopamine detox. The clinical data entirely rejects this concept. It is biologically impossible to acutely alter baseline receptor density through short-term deprivation. You cannot fix a downregulated receptor network by simply avoiding screens for a weekend. Overriding this mathematical glitch requires an evidence-based approach, restoring top-down inhibitory control over the craving circuitry. This overlay demonstrates the exact structural requirement. The prefrontal cortex must regain the capacity to project strong inhibitory signals down into the hyperactive striatum to regulate its output. Rebuilding this neural network takes time and deliberate practice. It requires cognitive behavioral therapy and strict physical control over your environment to permanently remove the variable digital cues that trigger the prediction error. Pathological wanting is a structural, mathematical failure of the brain's prediction hardware. You cannot out-willpower the math. You must systematically eliminate the variables from the equation.